going to be talking about transcription in this video. Here's a slide of some vocabulary we need to know. So we're going to first start off with DNA. We're going to get our core enzyme and our sigma factor. The RNA polymerase is going to get directions from the sigma factor because it knows where to find the promoter. The promoter is seen here at the -35 and -10. The promoter is also going to determine which strand is going to be the template and the non-template. So in this case, our blue strand is going to be the template. Our transcription bubble is going to have to form by unwinding the DNA. It's going to look something like this. Our Tau box is relatively easy to break because it has Ta bonds, which we know have 2 hydrogen bonds, and our Cg have 3, so it's easier to break those. We're going to do a couple of bonds with both the holoenzyme and the RNA polymerase, and this is going to be known as our +1 region. Our coding strand and our RNA strand are going to be relatively the same, except we know that RNA deals with U's rather than T's. Our phosphodiester bonds are going to connect the backbone of the strand. So right here you can see that they're connected here, and then our di obviously means 2 ester bonds, and then our nucleotides are going to be over here. The sigma factor is going to detach after about 9 to 12 bonds have been made, and it's going to leave the core and the RNA polymerase to do the work. Elongation is going to occur 5' to 3', so we're going to add on this end of the RNA, and in this example we are going from this way and we're going towards the right. I've also noted it down here on the left, just keep that in mind. So there's going to be 2 ways to terminate. We either need this RHO factor or we do not need it. So the RHO is going to unwind the RNA from the DNA, and you're going to end up with these 2 products right here. Otherwise, it's going to be inverted repeats, which is going to be a lot of C's and G's that have bonded together. Because RNA is kind of folded back on itself. The DNA is going to have a lot of A's after this has happened, and U's we know bond with A. As I said before, these are relatively weak bonds, so the RNA polymerase is basically going to fall off because this is not as stable as it could be. There's also head-on collisions, which make premature terminations, and these are not good. So after a terminator has been transcribed into our sequence, that basically tells us that transcription is over, and this can be translated into proteins or whatever it needs to be. If we were to add our 5 base pairs in between these, it's either going to be this area is not going to be 35 base pairs ahead of transcription, or this area is not going to be 10 areas ahead above transcription. So basically this amino acid is not going to get transcribed or translated because the sigma factor doesn't know where to bond because it's used to bonding right here and right here. So if these were to move anywhere, it's going to not recognize that. So anything this amino acid was needed for is no longer going to be in sufficient amounts. But there are weird cases where transcription does occur without the sigma factor, and the RNA polymerase will actually translate random points in the DNA, which has a very high chance of causing mutations.